Good evening, everyone. Thank you so much for joining us for this Zoom In event focusing on plant science and sustainable agriculture. Joining us this evening is Dr. Alex Harkess. We're so glad that Dr. Harkess has joined the HudsonAlpha faculty. He not only holds a joint position as a faculty investigator at the HudsonAlpha Institute for Biotechnology, but he's also an assistant professor with the Department of Crop, Soil, and Environmental Science at Auburn University. Harkess joined Auburn and HudsonAlpha in 2020 after 4 years with the Donald Danforth Plant Science Center in St. Louis. He was a National Science Foundation postdoctoral fellow, and he focused on research looking at evolution, the function of small RNA pathways, the evolution of sex chromosomes in plants, and broad-scale comparative genomics. Dr. Harkess earned his PhD from the University of Georgia and a BS in botany from Miami University. And we are so glad to have him now in the great state of Alabama. We live in one of the most biodiverse states in the country. It is one that those who are aware of its amazing treasure call it America's Amazon. And so we're so thankful to have Dr. Harkess and the passion and the experience that he brings in agricultural science. And so in a nutshell, Dr. Harkess, can you tell us what your lab's current research interests are?
Sure, yeah, thanks for having me. This is fun. My laboratory studies two major things. We study evolution of plants and we study how plants reproduce. So on the evolution side, we look back in time, almost 1 billion years ago to when the first green thing evolved on the planet, all the way to today, where there's almost 400,000 some plant species living on the planet right now. And so we get to use genomics, for instance, doing DNA sequencing at the Institute here to build a library of all of these different plant species to tell stories about how they've evolved and changed over time. On the reproduction side of my laboratory, we're just fascinated by how plants reproduce, how the flower works at all, how it makes pollen and eggs so perfectly every single time, and how you can start to control those different aspects of plant reproduction to maybe improve hybrid crop breeding to make more food on less land for people like the state of Alabama.
That is such an incredible goal to think about as we think about the growing population of our planet and the amount of land that we have to be able to farm. I'm going to go back and talk a little bit more. You talked about sequencing plants. Can you tell us a little bit more about what it means to sequence a plant and how you guys go about doing that?
Sure, yeah. Just like in humans, every single cell of a plant contains DNA. 4 nucleotides define so much about how a plant looks and grows and functions over time. These 4 letters, A, T, C, and G, are the nucleotides of DNA. They are the building blocks of sorts that make up genes that get stacked into pieces that make longer bits that end up as chromosomes, right? And in humans, for instance, in just about every single cell of our bodies, we have 23 pairs of chromosomes. Plants do things kind of similarly. They just have different amounts of DNA and different genes and sometimes lots of chromosomes. And when I say DNA sequencing, how do we get that information out of a cell? How do we actually take chromosomes out of a cell, whether it's a plant or a human or anything else, and actually read a chromosome? And Jane Grimwood in particular and Jeremy Schmutz have been doing this their whole careers and have really built a mecca here in Huntsville, Alabama to do this at huge scale, not just with plants and not just with animals, but all sorts of organisms across the tree of life. So we can quite literally take chromosomes out of a cell, break them up into pieces, and read those 4 letters over and over and over again to almost fill a library with new books that tell stories about how plants have changed over time.
Thank you so much. And this is a great chance that if ladies and gentlemen of the audience, if you have any questions, feel free to type them in our Q&A. And throughout the evening, Dr. Harkess and I will kind of look through those questions and we'll take a pause kind of about midway through and really go through and have an opportunity to answer some of those. And then again, at the end, we'll come back out. But please feel free to type them in as we go through. So when you talk about the research that you do, what initially led you to this passion to be able to pursue this particular area of expertise?
When I was 18 and about to head off to college, I thought I was going to be a lawyer. And I'm so glad I'm not, because turns out I'm terrible at following the rules, which is kind of a key component of being a scientist, is that the rules kind of need to not exist in your life, right? You need to break all of them. You do the illegal
Super
things.
creative, yeah.
Exactly. Yeah. So when I got to college at Miami of Ohio, on my first day of college, I met a botany professor. And he sat me down in his office and said, here's what I love to do. I study ferns. I've found new species of ferns in the Bahamas. The reason I love being a botanist is that you get to tell a story that has never been told before. And we get to do that with the way plants look, and we get to use DNA and RNA sequencing, and you get to quite literally write chapters of the history of our planet, something that no one has ever done before. So every day, in a sense, you get to discover something new about a planet that has been around for billions of years now. I found that incredibly charming. So that day, I changed my major to be a botany major. My first semester, I was kind of flailing a little bit. I didn't really know what I wanted to do. And a TA of one of my courses said, why don't you come to my laboratory? I'm a PhD student. We're working in a really cool lab doing sex chromosomes in papayas is kind of the focus of our lab. And I thought, sure, might as well show up. So I get into that laboratory and I start working. And all of a sudden, I become immersed in this totally different language of DNA, a totally different branch of the tree of life, plants, that I didn't really know that well. And I became fascinated with how flowers work. Papayas is kind of a fun story. If you don't know anything about papaya, it actually has 3 sexes. It has males, females, and hermaphrodites. Males are kind of like humans. They have an X and a Y chromosome that make them male. Females have 2 X chromosomes that make them female. And hermaphrodites are XY male with a weird Y chromosome that makes them produce hermaphrodite flowers that have both male and female parts in every single flower. They can self-pollinate that way. But what's interesting is that papaya breeders don't want fruits that come from hermaphrodite flowers. They taste terrible. So they only want fruits that come from female flowers. So a key goal is, how do you get rid of hermaphrodite flowers in a population? How do you control the sex of a papaya plant? And it turns out it's not just a cool scientific story, it's worth a ton of money. Right? Breeders don't want to be growing plants that make terrible tasting fruits. So how can we use genetics and genomics to try and figure out how to make these plants taste better by controlling the sex? And that led me down just a really amazing scientific career path where I did a PhD studying sex chromosomes in asparagus plants. So when you go to the grocery store, asparagus actually also has sex chromosomes. XYs are male, XX is female. And same story, actually. XY male plants taste better and live longer in the field. And breeders don't want XX female asparagus plants. And the problem is that you don't know whether a plant's going to be a boy or a girl until it flowers. Or at least, a while ago, we didn't know that. And breeders would have these plants out in the field for years, 3 or 4 years before they'd make flowers. And you'd find out that, oh, this 50% of my crop is females, I'm going to rip it out of the ground. And then you've wasted half your field space and a ton of money that could have been made. So in my PhD, we started figuring out the genes that control sex in asparagus plants. And now, using just a couple of genes that we can modify, we can control whether a plant is going to be male or female or hermaphrodite or nothing at all. And turns out, again, that's worth a lot of money and changes the landscape of plant breeding. So it's a really cool evolutionary story, which is a real big passion of mine. How do sex chromosomes evolve in the first place? How do separate sexes of plants evolve at all? And then applying that to plant breeding is really important and economical and valuable for how breeders actually make crosses and develop new hybrid crops.
What a great story, Alex. And I'm trying to make sure that I've got this correct. So we said female papaya is the goal, and then male for asparagus.
Yep.
So are they currently using genetics tools in the growth and development of those crops to ensure that we have more male and more female papaya and more male asparagus?
Absolutely, yeah. A lot of companies now are able to do DNA sequencing of those sex chromosomes and figure out, from even a seed, is this seed going to grow up to be a male plant or a female plant? And now we can do that using tools like these DNA speed readers, these DNA sequencers that we have a bunch of here at the Institute.
What an incredible amount of time, effort, energy, input that's saved for our agriculturalists. So seeing that immediate feedback from the work that's happening in your lab, just from your PhD research to now what we're seeing utilized in the field, and to imagine what that's going to look like in the future. As we, one of the questions that we had as we were talking about that was, someone asked if you have done any work with mushrooms, if you guys have sequenced or done any work on mushrooms.
Interesting. Me personally, no, not at all. I think mushrooms are totally fascinating though. There are some really exciting projects going on. You know, there is, for instance, 1,000 Fungi Genome Project that is, I think, largely funded by the Joint Genome Institute, Department of Energy plant arm, which Jeremy Schmutz and Jane Grimwood play a huge component of. So I think they're actually leading the charge in developing huge genomic resources for fungi in general. Me personally, no, I don't do much with the shrooms at all. I think they're fascinating though.
I agree. Just all the different variation that exists within them, they're incredible. So if you're not working on mushrooms, but you've talked about papaya, you've talked about asparagus, what are some of the other things that your lab is working on? What is one of the most recent findings that you're the most excited about?
So the fun part about being a botanist who has access to all of these DNA and RNA sequencing tools is that we get to ask questions first and then find the best system, the best species to study those questions in. So one really kind of fun one lately, about 2 years ago, a professor from Stanford, Virginia Walbut, who's a very famous corn researcher, studies how corn tassels, the male parts at the top, kind of develop over time. She was a hobby dahlia grower. And you know, she asked me, Alex, are you interested in dahlias at all? And of course I said, not really, not especially. And she started sending me email after email and photo after photo of amazing dahlias. And so I got fascinated by diversity of horticultural crops. And the American Dahlia Society gave us quite a bit of grant money, actually, to start doing DNA sequencing of dahlia flowers. So now I'm entrenched in this amazing dahlia world. I went to the national show last year, or 2 years ago, I guess, and I walk into the showroom, which is 1,000 dahlia flowers from hobby and professional breeders all across the country and even the world, actually.
Oh, that was stunning.
It was amazing. And I got to see something that kind of blew my mind. You walk into a room, and for someone who studies flowers and the diversity of flowers, I saw so many different shapes, forms, colors, and smells of different flowers. And these breeders, their competition works this way. They have 20 different categories of dahlia flowers. Some are really big palms, which are giant dahlia balls that can be really huge, the size of your head, or some that are smaller that come in multiples. Some dahlias look like water lilies almost. They're like kind of flat. Other dahlias have these beautiful curved petals. Some have flat petals. And I started looking at these flowers and seeing common flower mutations that we see in other kinds of plants. All of a sudden, I realized a horticultural society actually had one of the most diverse kind of resources of incredible plant material that could have existed. So what we're doing now with the American Dahlia Society is I have a master's student that I met at that meeting, actually, who's going to be sequencing a dahlia genome and then resequencing the best in show flower winner from each of those 20 categories to start mapping the major mutations that underlie these different floral forms. And maybe along the way, we can start to figure out how to save horticultural societies by injecting young members into them, by bringing genetics and genomics into them, and to study the problems that plague a whole lot of horticultural breeding, like viruses, for instance.
Oh, I should have talked to you before. I just planted dahlias in my garden and anxiously waiting for them to come up. I'm anticipating the large head-sized ones, but we'll see what happens. One of the things that you mentioned, and I saw you get so excited about when you're talking about the dahlias, is the amount of diversity that exists within the dahlias. Can you talk a little bit from an agricultural, from a plant perspective? Why is it so important to have a lot of diversity within a species?
Oh, that's such a great question. The word diversity is a huge word to unpack. I like to think big first. So there are almost 400,000 species of plants on the planet, right? And so when we think about what plants have been through in their roughly 1 billion year history from when the first green thing evolved, it's amazing. Plants have survived meteor attacks that quite literally, the dinosaurs went extinct, except for the lineage leading to birds today because of some of those attacks.
And yet plants can't move.
Plants can't move, and yet they survived, right? On a planet that is often trying to kill them. And that was before humans, even showed up, right? And we've caused all sorts of other kinds of problems too, that plants always figure out a way to adapt and evolve and survive. And I find that amazing. So, a key aspect of finding solutions is to look on the planet at species that have already evolved certain adaptations to survive. And then using DNA sequencing, you start to understand the genes or the mutations that make some plants able to survive in different situations. So there's kind of one aspect, right? We look and we survey what already exists on the planet today. At a very fine scale, we do the exact same thing with plant breeding. You can't breed a crop with just one plant, right? You need to have diversity that exists. You need to have different kinds of mutations that confer different kinds of benefits, because we still don't know what genes do for the most part. And we're kind of guessing a whole lot of time. Even, this is the same problem even with human genetics. We say that, one gene, does something, and a lot of times we actually don't know that. And so plant breeding is really an immensely complicated and beautiful dance. And people like Josh Clevenger, a new faculty member here, are the experts at doing this, especially because people like Josh's lab can understand how to use DNA and DNA sequencing to look first at the diversity of what exists for a given species, and then two, to start making thoughtful crosses between plants and then sequencing the offspring, and then doing this over and over and over again to understand how DNA changes over time through the breeding process, and how you can pass along beneficial genes or beneficial alleles or copies of genes to the next generation. But it all starts with diversity.
I love that. I was just recently reading about biodiversity and how E.O. Wilson coined that term, who's an Alabama native, was one of the first to coin that term in the mid 80s. And so just as we continue thinking about diversity and all the different applications of that, you mentioned the Dahlia project. Can you tell us about another project that your lab is working on, another passion that you have?
Yeah. I've got, there's a bunch, right? This is the fun part about being a botanist, is that you could go on and on and on about cool stuff happening in the lab. There's one that really makes me excited lately. About 2 years ago, maybe 4 years ago, the kind of every graduate school in the country decided they were going to get rid of the GRE, right? The graduate exam that you kind of had to take to get into grad school. It was expensive. There was, this huge push to reduce barriers to entering grad school, which was fantastic. But what we didn't realize is that by doing that, we changed the goalposts a little bit, and that now graduate admissions valued different things from undergraduates who are applying to PhD and master's programs. So when I asked a first-year PhD student, hey, what do you think was, the thing that made you most successful in your application? And this person said something very interesting to me. Oh, it's because I had publications as an undergraduate, which blew my mind. I didn't realize people were publishing as undergrads. And apparently that is now, the new kind of gold standard for getting into what some people perceive to be, top PhD programs or their dream schools or something like that. And so it made me think, well, how do we reduce those barriers for entry for students that can't work in laboratories enough to get publications as undergraduates who need to work in dining halls for money, for instance, right? It's this clear socioeconomic barrier that we didn't realize would happen when we took the GRE away. So I started at Auburn about a year ago. March 1st was my start date, which was a weird time to start a new job. And Auburn has this, really rich history, of course, of sports, of plant breeding, they have this old rotation, which is the longest ongoing crop rotation that exists. And then people talk about this really sad story, which is the Toomer's oak tree. And these trees were about 100-something years old. They're downtown in Auburn, Alabama, right by the Toomer's drugstore. Students would roll toilet paper over them after football games. And about a decade ago, someone poisoned them tragically. A rival football fan, if you want to say fan, I don't know if that's the right word. Totally killed them. But luckily, professors from the horticulture department at Auburn saved perfect clones from that tree. And one botany professor put one in his front yard and didn't tell anybody for a decade. And so I started thinking, now that we have these amazing genome sequencing tools here at HudsonAlpha, what if we could reduce barriers for students to enter publishable academic science? What if we don't have to have undergrads working in laboratories to be able to do publishable research? What if we could bring it to them via courses? So we did a crowdsourced fundraising event through the Auburn Tiger Giving Day and raised almost $11,000 to sequence the genome of this clone of the tumor's oak tree. And now in fall of next year, I'm designing a flipped classroom undergraduate and graduate level class where students in my course are quite literally going to be assembling this genome of the tumor's oak tree that is so important to their campus and publishing it at the end of the semester, so that all students in this course will have a manuscript, right? They'll be authors on something. So no longer, do we have to, rely on these socioeconomic barriers that limit some students to not being able to enter graduate science.
I love that. We've so firmly being a part of the education outreach team here at HudsonAlpha, and it's such an important thing for our students to be able to get those experiences and to be able to break down those barriers. And so we're so thankful we can't do what we do without having researchers like yourself to mentor and to bring those students along, and giving them that experience is just phenomenal. And from an Auburn alumni, I am just incredibly thankful and was totally over the moon excited when I found out that you guys had gotten funding for that project and we're going to be able to take that on. And just thinking about the fact that the genetics and the genomics technologies that we have can make this possible, that these students are going to be able to do this, that it's accessible to them. It's not just something that happens in the research labs. It's something that they can understand. And as we look at the students coming up in the next generation, helping them to understand the role of bioinformatics and computational biology, that it's not all wet lab, and giving them a really real perspective of what the field is going to look like, and hopefully igniting the passion for them, like you had with the professor with ferns and with the papaya, that this will light that fire in them and can encourage them to solve their own questions. I love how you talk about being able to ask all the questions, and seek to find those answers. And with botany, with agriculture, you just have literally a field of questions that you're able to answer. So we had a question to come in from the audience, and while we were talking about that, and they said, what kinds of things are going on in agricultural genomics that might be able to help your everyday home gardener?
Very interesting question. It's something we actually a lot of people don't talk about this, right? We only try to think about, row crops, feeding the world, that kind of stuff. And that's not the only thing that kind of matters in the world. I wish I could be an avid gardener. My dog eats everything in the backyard. And so I am dying to get better at gardening once I can, cordon off an area or something. I guess my best response to a question like that is everything we learn about a plant, from what a gene does to how plants physiologically work, right? How they soak up water and how they soak up sunlight and make energy out of that. Every little bit of that knowledge informs us. And so, for instance, even the home gardener uses fertilizers, right? We all use a lot of water in some cases. So what if we can start to apply knowledge from row crops, right? We're making all these amazing advancements and making plants better. Will those trickle down into horticultural crops? Absolutely.
I think you have given hope to tons of gardeners and hopeful gardeners with green thumbs everywhere as we think about the importance of those lessons that are learned, and from a food crop, from that standpoint, thinking about all of the benefits and the promises that can come. So one of the things, thinking in those terms, where do you see this the field of sustainable agriculture and plant science moving in the next decade? What are some of the promises that you see, and what are some of the challenges that you think this field will incur?
So I'll speak specifically kind of from what my lab does, which is maybe the fun thing. I love studying reproduction, and it's kind of like, I told these stories about how, studying papayas is really exciting and asparagus is really exciting, but they have real world implications. A lot of what my lab does now is trying to understand exactly how flowers work, right? How can we control them using all these new amazing tools like gene editing, for instance? So I think the future of the field, at least for plant reproduction, is the ability to make new hybrid crops. And if you need a crash course on what hybrid crops are, there's this really funny phenomenon called hybrid vigor. Sometimes when you take two very different plants of the same species, and by very different, I mean they look different, they came from different places, they're very genetically diverse, and you cross them, sometimes the offspring of that cross are bigger and better than either of the parents are. And we totally don't understand how this phenomenon works, but it's so important to plant breeding because you can make more food than either of the parents ever could. And so companies keep this information very close, of course, the parents of a cross that make a really good plant. And that's why, for instance, breeders have to buy, or farmers have to buy new hybrid seed every single year, and you can't plant one of the parents. So what I think, the future holds is how can we make hybrid crops out of species that don't currently have any hybrids. Like soybean, for instance, is a really funny example. Soybeans are cleistogamous. They do this really funny and obnoxious thing where before the flower even opens, they pollinate themselves. So if you're a soybean breeder, you have to put on your specs and you have to rip open this tiny, 1 or 2 millimeter flower bud under a microscope, rip out the male pollen before it pollinates the female organs and then makes a seed. And then you take that pollen and very quickly run over to another plant, rip open a flower bud that's very, very tiny, start jabbing the pollen in it. And that's how soybean works. Well, what if we could use our genetic knowledge and start to control how a flower functions? Make soybeans that are males or females, make soybeans that can be crossed in a field with bees instead of laboriously by tweezers, under these tiny microscopes. I think that's where my lab can contribute to the future of sustainable agriculture. Can we make new hybrid crops where they previously didn't exist?
Fantastic. And that leads us to a question that we got from one of our attendees this evening. He talked about, he's asking about heirloom plants versus GMO plants. So I think kind of extending a little bit that hybrid and gene editing conversation that you had, he was asking your opinion on some of the pros and cons for both of those.
That's a really big question. That's probably one we'd have to hang out for a full day to talk about. There's a lot.
Agreed.
Delicious heirloom varieties of stuff, and an heirloom.
Cherry tomatoes.
Absolutely. Yeah. And heirloom is a funny word, and no one really knows what heirlooms are anymore. They're just older varieties that were, maybe stuffed in someone's pantry for a long time that are kind of getting resurrected now. And a lot of those taste delicious. Some don't perform very well in different environments. Other commercial varieties of tomatoes, are bred to grow very well in different places. And those aren't GMOs, by any means. Or they don't have to be. And so I guess, that's a really big question. My suggestion is if you can grow it in your backyard and you can eat it and it's delicious, plant whatever you can and whatever you want.
I love that. Talking about getting a garden in the ground, that was my accomplishment yesterday evening. I got some Brussels sprouts and beans and radishes and flowers all ready to go. And so I love being able to think about all of the different things. And I know as you were talking, one of the things you mentioned earlier is really using genetics to inform breeding, being able to figure out what traits a plant's going to have, to be able to reduce the amount of time, effort, and energy that it takes in a crop. Instead of having to grow half a crop and then rip it out, you can use those genetic tools to really be able to advance that. And I think that's a really powerful tool. One of the other questions that came in while we were chatting is, have you had an opportunity to see the Huntsville Botanical Garden Trillium population yet?
I have been to the garden twice now. I met Tracy Cook, the lead curator there, and
It's
I
fantastic.
mess up the first name. Green is her last name, the CEO of the garden right now. And I love botanical gardens for a bunch of reasons. One, they are the most amazing repositories of plant diversity on our planet. If you believe in saving libraries to continue education for young children, you should also believe in saving botanic gardens for the exact same reason. They are the kind of the bastion of how we preserve plant diversity on our planet. So there's my botanical garden soapbox. I think what the Huntsville Botanical Garden does is amazing. I've been out there and I've seen their collections, not just of large trees, but of horticultural flowers. I've seen their azalea collection from a breeder that was there for almost 35 years, it seemed, making hundreds of thousands of different crosses of different azaleas. The Trillium collection, I haven't been there in a while. So maybe after this, I'll send Tracy Cook an email and see if I can drop by. There's so much amazing stuff. When you walk into a botanic garden, you should be thinking about how you're looking at hundreds of millions of years of history right before your eyes. And where else in the world can you do that?
So few places. And speaking of history, one of our participants has asked, what do you expect in terms of breakthroughs in the next 10 years?
Oh man, if I knew that, I'd be in the stock market right now. That's a great question. We've had our minds blown, it seems, every year with new technologies. And there are so many brilliant people studying so many different questions in so many different plant species where that is a hard question to predict. And a lot of being a scientist is being able to be surprised and adopt new technologies as they get thrown at you very, very fast. People at Hudson Alpha, like Kankshita Swaminathan, are particularly good at that. Her laboratory studies gene editing, right? And new techniques for gene editing. You might have heard of CRISPR or CRISPR-Cas before. Those have been out for, 6-7 years now or something at this point. And they just keep on evolving. Every day, it seems, we have new ways to edit a plant, to change the sequence of a plant's DNA. And that is opening up totally new doors for how we understand what genes do, how we can make plants better in the field or in the laboratory, and how we can use plants for different reasons, like maybe even plants as factories for different things.
Speaking of plants as factories, this is a perfect question. I know I have heard you talk before about duckweed. And duckweed potential. And so one of our attendees has asked about duckweed having biofuel potential. And so if you could talk a little bit about biofuel, about duckweed, about biofuel, and a little bit about your interest in that area.
Oh my gosh. How much time do we have the rest of the night? Yes.
I know, right? It's 6:37. So
I have duckweeds growing in my windowsill. I have duckweeds on every ledge that gets sunny in our house. My wife, she's ready for it to all go to the laboratory. Duckweeds, if you don't know what these are, you've probably all seen them before on the tops of ponds. Duckweeds are aquatic plants that are the fastest reproducing plants on the planet. They can divide from one plant into two in a matter of 12 hours for some species.
wow.
So that means they can reproduce at a rate greater than any other plant on the planet. And they can do it without needing any soil. You don't need to plant them. You can grow them on wastewater, wastewater from farms, for instance, even wastewater that has manure in them. Duckweeds love that. So yes, duckweeds have huge potential as a biofuel. And we're currently trying to figure out ways to make these plants grow even faster. And not just for biofuel, actually. We think a lot about, we put so much fertilizer on agricultural fields right now. And where does it all go? It eventually runs off and goes into the water table. So how can we start to accumulate that nitrogen and phosphorus from fertilizers that is a finite resource, actually? We have to mine these kinds of things out of countries. So duckweeds are really neat to me because they are called hyperaccumulators. They're kind of like sponges, actually. So they sit on the top of the water, and whatever's floating around them, in terms of nutrients or anything like that, elements, duckweeds suck them up. Even heavy metals like titanium and lead, duckweeds just pull up into their bodies, into these little air spaces around them, and doesn't bug them at all. So I look at that and think, geez, well, what if we could start to use duckweeds, for instance, as a way to hyperaccumulate nitrogen and phosphorus out of wastewater and separate it out and make renewable fertilizers? What if we can use plants and reshape what a weed means? You ask a golf course owner what duckweed is, they're all going to say, yes, I hate it. It's the worst plant on the planet because it clogs up our water pumps and it gets into our lakes and it makes the lakes kind of, look planty. And for me, I look at a duckweed and think, oh my gosh, it has evolved an incredible adaptation to grow so fast. How can we use that adaptation in a very different way than some people might perceive? A weed is just a plant that hasn't learned how to grow straight in rows yet, is kind of what the farmers like to say.
That is fascinating. I love the fact that the duckweed really does change your perspective when you think about them being hyper accumulators and all of the different potential that you can have from that. Another question that we had from our audience is, and I love this, what does your field lack besides better funding? And we always, we all know that Hudson Alpha is a nonprofit organization, and we are so thankful for all of the incredible support that that you guys continue to give us through the years. And so we are always thankful for that funding. And so we're always welcome contributions to the Agricultural and Sustainable Science Fund at Hudson Alpha. But with that, I will ask Alex, what else does your field lack? Where are those growing edges?
I'll say two things. The first, it's been really eye-opening being at an institute that started out as a human genetics research institute. And human genetics has so many amazing tools and funding and resources and infrastructure built around a single species of mammals. And you come to the plant world, and the 5 Hudson Alpha plant faculty, together between us, we probably study 100 plant species or something. I think we've sequenced hundreds of plant species, worth of genomes at least. And so that adds so many interesting complications, because whereas human geneticists, for instance, have these great tools and infrastructure to study one species, and all those tools work very, very well for the most part. With plants, every new species, every new variety we look at, it's often different, and things don't work the same. So we need better infrastructure, better tools that work on multiple species. That's been a key complication. And I'll say the second is the field of agriculture in general lacks diverse representation from all groups of people. And this is something that Hudson Alpha is actually specifically addressing. Myself and Neil Lamb and Kangshita Swaminathan are co-PIs on an NSF grant called Includes, which is meant to increase representation in the sciences. So this grant is specifically for us to look at all the root causes across Alabama in particular and ask the very simple question: why is the field not as diverse as it could be? What are the institutional barriers that have limited some people from entering the field? And then second, how can we build better connections between all these excellent institutions that already exist? Boys and Girls Club, 4-H, Auburn University, Alabama A&M, and so forth, to accelerate students from all groups, all socioeconomic groups, to become plant scientists. How do we remove those barriers? So I think there's a little bit of science, and then there's a little bit of social for what the field is lacking. But luckily, Hudson Alpha is very thoughtful and doesn't just think about the present. The conversation is always eyes down the road, thinking 10 years in the future. What will the next set of advancements be? What will be the problems in the field? And everyone recognizes that the problem in the field is educating a STEM workforce and making that a diverse workforce too. We talk about diversity being so important for plants. It's equally important for the scientists who study them.
What a beautiful perspective, Alex. Thank you so much for sharing that, and for being a part of the solution to be able to figure out how we're able to bring in more perspective and more thought and more creativity and questions. And I love the fact that I get to work alongside of you guys and watch these programs grow and be able to encourage people to pursue these fields and to know that there are opportunities for them to do so. You were talking about, as you were talking about that, I was thinking, there's so many great mysteries in agriculture, in your field. If you could solve one great mystery, what would it be, and why that particular puzzle to solve?
That's an awesome question. I love what's that quote? If you want a new idea, read an old book.
Yes.
I love reading old literature from people like Charles Darwin, who just looked at plants and thought very, very deeply about them. In a letter in the 1830s to Joseph Hooker, Charles Darwin described the explosion of diversity of flowers as an abominable mystery. And that is a quote that is stuck with all plant biologists in their career. Why did flowers evolve to be so diverse, right? You'll find flowers of all different kinds, shapes, sizes, across all different species on almost every inch of the planet. The way they look, grow, and smell is so incredibly diverse. And that all happened very, very fast, actually, in a short period of time. We think the first flower evolved maybe 130 million years ago. And the greatest mystery to me is, what did that first flower look like? What did its genome look like? What genes had to change or mutate or double or something like that allowed the first flower to evolve? And we can't go back in time to answer that question. So what we do is kind of like what I've talked about now, is we write the story ourselves. We sequence DNA from plants all across the tree of life. And once you do that, you can start putting those puzzle pieces together and going back in time to ask a very straightforward question. What did this plant look like maybe 150 million years ago when it first evolved? You get to be a time traveler. So I think if I could solve any mystery, I would solve Darwin's abominable mystery.
I love that. An abominable mystery, thinking about all of the incredible diversity. And I just, as I picture that, I just picture this beautiful field full of all of the flowers and all of the contributions that those make, and thinking about how that has just kind of been the thread through this evening's conversation, is the power in diversity, both having diversity among the dahlias and the species, diversity in individuals bringing in new perspectives, diversity in collaborations and partnerships, and just having all of that together, being able to learn from that, and thinking about all of those different pieces. So I have in addition to that, I have a question that has just come in relationship to the shortage of bees and plants that require pollination. So do you have any information about kind of the impact that that's having on plants that require pollination?
Yeah, that's a great question. And that is also a huge one too. There's people all across the world, addressing this in different ways. And it also depends on what plant you're working with. A soybean, for instance, pollinates itself. You don't need bees out in the field. Corn plants just need a little bit of wind. You just gotta kind of hit them really hard most of the time, and pollen falls down. So it's really dependent kind of on what crop you're working with. What to do about it? Oh, geez. I think a lot of these problems, one, start with addressing, the key problem, the key problem areas, which things like pollution are huge ones that drive bee shortages. What to do about it is a good question. I don't have a great answer, actually. I think it's something that we need to keep on considering. I think understanding how flowers function is going to be a key aspect of that. Maybe we can start to develop new plants that, for instance, might not need pollination by bees, that could be wind pollinated or could just be self-pollinated. That's certainly not off the table.
I love it. So one of the things that we had the pleasure of seeing this week, we were on a team meeting, and we got a virtual field trip, and we got to see some of the amazing new grow rooms that are in the 701 building on the HudsonAlpha campus. And they are just fascinating with being able to control light and temperatures and daylight. And can you tell our audience a little bit more about those brand new growth rooms, and about the dreams and the visions that you have for the things that will take place in those rooms?
It's a really exciting time to be a plant biologist in Huntsville, Alabama. And this place, for instance, has long been a mecca for human genetics and human genomics. And now we are at capacity to just completely be so successful in plant biology and plant genetics, because Hudson Alpha has invested a lot of resources for us to really study plants. And part of that is being able to grow plants. Plants are actually not very easy to grow. They need a lot of requirements. They need a lot of energy and a lot of time and a lot of water. So about a week ago, we just unveiled the opening of about 1,600 square feet of very high quality plant growth space in the building next door to the main building on campus. We have 5 individual growth rooms that are controlled by these really amazing lights, where we can perfectly program exactly what wavelength and what durations of different frequencies of light that we want and what colors of lights we want. And now we're just growing all sorts of stuff. We are off to the races. So Josh Clevenger's lab, for instance, is doing some really amazing stuff by trying to speed breed peanuts, which is when you just confuse the plants by keeping the lights on 24 hours a day, so they grow really fast and flower quickly. You can squeeze in more generations per year, as a breeder. My laboratory is growing stuff in one of our rooms. Josh Clevenger, I think this is hilarious. He calls it the room of wonders, because my lab studies so many diverse plants, and this is kind of the fun thing. We have some fast flowering mini maize growing right now. This is a 2-foot tall variety of corn that flowers in about 3 weeks from when you plant it. So compared
I cannot wait to see those. We have to get pictures out when you get those. It said 3 weeks from seed to corn.
Yep. So you get a little baby corn cob, a few weeks later, that you can, actually try and, test some breeding ideas in this super fast flowering corn variety this way.
What kinds of questions will you be asking?
Oh, geez. So, we're fascinated by how perfectly male parts, the pollen grain gets formed. And so it's really, you can do this in, tall corn out in the field, or you can squeeze many generations in per year in kind of these short stature varieties of corn. In particular, I'm fascinated by how pollen grains form so perfectly every single time, right? It's the male gamete, the male part. You need it for every single successful fruit that gets formed. So now we can study corn almost twice as fast, basically, by being able to grow these short stature, fast flowering kinds of varieties. We're also doing some really neat stuff. I have a wonderful postdoc in our laboratory, Dr. Sarah Carey, who's become fascinated by hops. Hops are the cones that flavor your beer, for instance. They smell amazing, but again, only the female flower is useful, right? That's the cone that actually forms. So what she's doing is growing a ton of different hops that actually have different sex chromosomes entirely. They have different genes that make them male or female. And now she's starting to understand and sequence genomes of these different species of hops that have different flavors and everything like this, to try and understand, what is so special and diverse about the female flower of a hop cone.
And the diversity in that, and everybody who thinks about the qualities of hops and the microbrewery industry and all of those things and the value that comes from having all of that incredible diversity, not only in terms of the plant, but also in terms of enjoyment on the product side of that. We had another question that came in as we were talking about that, as we were looking at, and you were talking about Darwin's mystery. One of our attendees asked if, have any fossilized plants been sequenced?
Oh, that's such a cool question. This whole field is called ancient DNA. You can find, I think DNA has a half-life, meaning like half of it will be degraded by something like 5,000, 10,000 years, maybe longer. I can't quite remember. But you can extract DNA out of old stuff. We extract DNA out of herbarium sheets, and an herbarium is just a dead plant library, like with 4-leaf clovers pressed between books. We do that on a large scale. It's kind of like a library of old plants. And you can even extract DNA out of those that are hundreds of years old, some of which came from like Darwin and Linnaeus's journeys, on boats to go look at new plants. At the same time, people discover old plants in cool places, like old corn varieties in caves that were inhabited by early humans. at the beginning, the dawn of agriculture. And we can sequence those kinds of plants to understand, what did genomes of different plants look like, thousands of years ago, potentially, before very modern agriculture was a thing. In terms of fossilized plants, like, if you were to crack open a rock and see, a beautiful fern kind of inlay in it, I don't know anyone that's gotten DNA out of something like that. Most of the plant material and the DNA is totally degraded by that point. But that doesn't mean the fossil's useless. Fossils actually teach us a ton about how to calibrate our tree of life, right? Like if we find a fossil of a specific fern species and you can use carbon dating to figure out roughly when that happened, well, then all of a sudden you have a new kind of bookmark on your evolutionary tree of life timescale that says, well, this species had to have been around by this point in time. And it allows us to change our understanding of the tree of life, to make some branches shorter or longer, and maybe to change the locations of some limbs on that tree. So long story short, we are sequencing old stuff all around the world right now. And I say the royal we, people in laboratories across the world are doing this on all sorts of interesting species.
That is incredible. I recently found out that not only is Alabama have a wide amount of biodiversity in terms of life, but we also have an incredible biodiversity of fossils in the state, that it is kind of, it is on par with some of the Western states in terms of fossils, and that we have an incredible diversity and story that runs from the north to the southern part of the state, which gives us a lot of that variety in geology. So back again to the biodiversity and filling out all of those records. I know one of the things that we have both talked about, and then I want to make sure that our audience kind of leaves with tonight, is that there is so much happening. There is so much encouragement in this field. There is so much wonder for all of us to explore, whether it's in papayas or asparagus at the grocery store, whether it's in our garden, looking at local agricultural varieties, whether it's just in the world that we live in, making those observations that we get to kind of think about. I'll always remember as I look at that diversity of the flowers and thinking about the questions that those bring to mind in the incredible research that's taking place, as you've done such an incredible job of telling us, not only in your lab, but in the labs across the Genome Sequencing Center team with Jane and Jeremy and Kinkshita and Josh, that you guys are just an incredible team doing amazing work. And so I'd love to give you just an opportunity to kind of give us a closing thought or a closing statement. What would your biggest takeaway message be for us this evening? What would you hope that they would go and share with someone who was unable to attend this event tonight?
Oh, that's good. I think something that applies to plants and to people and to landscapes is that diversity is the foundation for change over time. And that could mean evolution and genes changing over time to make one species more or less adapted to another. It also requires that people change over time as well, to adopt new technologies, to be trusting of science, and to encourage and support scientific change as well. There's a lot of good in the world that's happening right now, and it all starts with just a little bit of diversity that exists and voices that are able to come together and enact change. And at places like HudsonAlpha, now we're doing that with diverse faculty, with diverse plants, with diverse questions. And now with all of the space to actually grow and study plants, we can truly enact this kind of change that not just benefits Alabama, but benefits the entire world.
Thank you so much, Dr. Harkess, for your time and for the amazing conversation this evening. Thank you for those words of wisdom. I'm going to close us for the evening again with that quote. I love, diversity is the foundation of change over time. So everyone carry that with you as you go through the rest of your week. And we hope to see you and hear from you again very soon. Thank you so much for the support of HudsonAlpha and all the research that we do. Thank you and have a great night.